Hello. It is already 12:05 and I'll have only 68 students here. Shall we get started? Can you quickly check in? The code is 6. You can see the check-in code 67331. 67331. Then we will start. Let me share my screen. Stop sharing. Hang on, let's share screen, my slides. All right. Can you see? Oops. Can you see my screen? Yes. Can you see my slides? Okay, thank you. So as I start, I will turn my camera so that if you watch your recording later, you'll not see my face popping out all the time. All right, so some of you have attended tutorial earlier this morning, and we actually went ahead and also discussed part of the second mycology chapters. So those of you who are in the Tuesday, Wednesday, and Thursday tutorial, we will have already covered all the contents in the tutorial questions. So again, this is the second lectures for mycology. I have uploaded the slides a bit earlier this time and also the recording. So just a very quick recap. Last week we talked about mycology. We basically look at the different types of mycosis, starting from superficial, cutaneous, subcutaneous, and then we look at systemic mycosis. So this is the different mycosis were divided based on the entry point of the fungi into human body. So the first three, superficial, cutaneous, and subcutaneous, very limited to the different layers or entry points, different targets in the skin. And for systemic, it is mostly of traumatic inoculation of environmental fungus into the subcutaneous layer. And in terms of the infection, it's usually more severe in terms of symptoms, but they are relatively rare compared to superficial and cutaneous mycosis. We also touch on opportunistic mycosis. So this is mycosis that occur when the immune response of an individual is weakened. So this can be individual who have other underlying medical conditions such as AIDS or HIV, or individual who are taking immunosuppression drugs. And we also have endemic mycosis. So these are mycosis that are restricted to certain regions in the different part of the world. So in terms of endemic mycosis, we talk about histoplasmosis. So these are certain parts of the US and part of South America and also Africa country, even in Asian country, those areas that has a lot of dust, also avian habitat, that there will be a lot of conidia spores that will be inhaled by individuals who then develop into different type of symptoms. But for today's the lectures, we are focusing on the virulence factors and also antifungals. So in antifungal has got two different parts here. Hang on, let me just go. Why is it not working properly? Okay. So first part, we're looking at virulence factors, a relatively small sections. At the end of this lecture, you're expected to be able to discuss the fungal virulence factors that contributes to host invasion, immune evasion, and persistence of fungi in the human host body. And we'll move on to second part in terms of the antifungal agent, medication that are used to treat fungal infections. We'll look at 6 different classes of antifungal and very briefly on their mechanism of action. How do they work? What is their target molecules? and what is the outcome of this antifungal agent. And you should be able to propose appropriate antifungal treatments for different classes of mycosis. So later on, we'll talk about certain type of antifungals that are mainly for systemic mycosis, and some are more for topical and oral routes of entry for the medication. And we'll then look at antifungal resistance. So similar to antibiotic resistance, antifungal resistance is also an emerging issues at the moment. So we look into, I think, two different major type of fungi that are now become antifungal resistant problematic. Hang on, let me just see. The slide is not moving, is it? Okay, let me try again. If I stop sharing. Hang on, stop sharing. Let me try and share again. Alright. Oh, I have two learning outcomes here. So now I'm sharing the first slide for fungal virulence factors. So as I mentioned just now, last week we looked at different types of mycosis. There are a wide spectrum of diseases that can be caused by this fungal species, whether it is superficial or to the more severe mycosis, such as systemic mycosis. And these virulent factors, just like what we talk about in the bacteria chapters, they are different genes or proteins that are produced by the fungi species that allow the fungi to persist and survive in the host environment that will then lead to diseases. And the challenges that faced by fungi in the human host body, including the challenges such as they'll have to respond to the immune responses in the human host, and also there are limited limited nutrients in the host cell, so it's not in the natural environment. The different part of the body may not have the nutrients that are ideal for fungi, and also oxidative stress inside the human host cell. So just an overview of the different fungal virulence factors. So this we'll go through 6 of these virulence factors one by one. First one is morphological transformation. For example, dimorphism. So dimorphism is a state of growth of fungi when they switch between yeast to mold or mold to yeast, depending on the different species that we're looking at. So why do they go through this dimorphism? We'll go into the details later. And also genetic and metabolic switching, adhesion via surface adhesion, secretion enzyme, cell wall component, and also biofilm formation. Are you not able to see the change of the slide again? For the slideshow, you need to share that window separate. So if I stay here, you can see, is it?
Yeah.
Okay, so if I go here, then all right. Okay, let me just enlarge this. This is already the largest. Then we go to the next one. First one, morphological transformation. As I mentioned just now, dimorphism is a switching between the different growth stage of fungi. It's either yeast or filamentous, which is mold, depending on the host environment. For histoplasma capsulatum, so this is one of the fungus mold in the environment that can cause endemic and also systemic mycosis. So in the environment, they form mold, but inhaled by human hosts at 37 degrees Celsius, they form yeast growth. So the yeast form is compared to our mold formation. The yeast enable the fungus to survive intracellularly inside the macrophages. So in a sense, going from the mold to a yeast form, they survive better inside the host. Okay. As for Candida, they can switch from yeast to hyphae structures. So the Candida albicans is a unique Candida species that we discussed briefly last week. It is the only Candida species that can grow as mold structures by producing hyphae. So the heavy structures can help the Candida albicans to invade tissues and also evade the immune responses. So typically the yeast forms, if you look at histoplasma capsulatum, the yeast formation, the yeast growth stage, help the fungi to spread in the bloodstream. So this is the advantage of yeast form. But the mold form, the hyphae structures, will then help the fungus to invade tissue and evade immune responses. So as conclusion, the filamentous form, they promote tissue penetration, invasion, but these form that are they are better for bloodstream spread dissemination. So this is just one example of Sporotrix kansky, one of the fungus mold that can cause what we call this subcutaneous mycosis. So on the depending on the growth form and the media that is used, you can see the mold formation on the image on the left, but yeast formation more on the brain heart infusion agar in the environment, 30 degrees Celsius. They can also go through genetic and metabolic switching. So fungi, they are quite good. They adapt differently in the host by changing the metabolic activities depending which part of the host that they're inhibiting. So for example, Candida albicans, they can switch to carbon, they can alter the carbon metabolism. They can use lactate instead of glucose in the oxygen's poor environment in the human host cell. And also, for example, Aspergillosis species, they can form siderophore, which is a type of protein that can help them to acquire ions inside the host environment. So they depend. What I'm trying to say is that depending on the condition in the host, these fungi can change the metabolic activities to enable their best survival inside the host. And also, they are heat shock proteins. So heat shock protein stress response genes, they are present in fungi, also present in bacteria and human. They are activated to counteract host temperature. So let's say we have a mold that is growing in an environment at 25 degrees Celsius. When they are in the human host, the temperature is slightly higher. So in response to that, they regulate the host heat shock protein expression to adjust to the host environment. So this is the second type of virulence factors. And also, just like bacteria cell, they can produce this protein, adhesion protein, that can help them to attach to the host cell. So if you recall the process of pathogenesis, for example, in other microbes such as bacteria, the adhesion is the very first step of pathogenesis. So just like that, fungi, they also have surface protein adhesion that help the fungi to initiate the colonization process. For example, in Candida albicans, they have agglutinin-like sequence. It's a group of protein that they will help to the Candida to bind to the epithelial and endothelial cell. For Aspergillus, they are able to bind to the fibrinogen and laminin through integrin-like protein. So different adhesion protein that we're seeing here that help Candida and also Aspergillus to attach to the host cell. And also, these adhesions are very important to help the fungi to form biofilm. So biofilm formation is not just in bacteria, but can also be observed in the fungi cell. And the biofilm formation can help the fungi to resist mechanical removal. So they are growing as a group of cells and attached very tightly to the host cell, and this can prevent mechanical removal of this fungi away from the host, and that form part of the pathogenesis process. And the next one, they also secrete different enzymes. So just like bacteria, the different enzymes can also form the virulence factors of fungi. So this enzyme can degrade the host tissue and inactivate immune factors. But these are the specific enzymes that we don't see in bacterial cell. For example, secreted aspartyl-proteinases, a group enzyme that breaks down protein, degrade the immunoglobulins, and also extracellular matrix proteins that help the candida to invade the tissues and the cell of the human host. And also phospholipases, like what we see in bacteria, this enzyme can disrupt the host cell membrane and help the fungi to invade the host cells and tissue. And in terms of reactive oxygen species that's present in the phagocytes, if you recall phagocytosis, within the macrophages, for instance, they produces different enzymes that will try to digest the fungi in this context. And so this is reactive and this is a very oxidative stress environment. So fungi counteract that by produces different enzymes such as catalases and superoxide dismutases. So this type of enzyme, you see that in the bacterial cell. So you'll see a lot of similarity in terms of virulence factors for bacteria and also fungi. And then the number 5 virulence factors include the antiphagocytic cell wall components. So if you look at the cell wall component of our fungi cell, they have beta-glucan structures. So in certain species of fungi, they have virulence factors such as the alpha-1,3 glucan that's present in histoplasma. They actually mask the beta-glucans from being recognized by the immune receptors. So these are the one way to help the fungi to protect themselves from being targeted. So if you look at bacterial cells, we have the toll-like receptors. But for fungal immune response, the common one is the dectin receptors is present in the human. So in a way to respond to that, they produces alpha-1,3 glucan to protect the beta-glucan from being targeted by dectin, the receptors. And apart from that, melanin, remember last week we talked about melanin in some of the fungal species. They produces brown and black pigmentation in the fungus. Melanin actually can help to scavenge free radical and also resist oxidative damage if and when the aspergillus are being targeted by phagocytes in this case. So the melanin has different mechanism in here. So two different roles. One is oxidative stress. The other one is free radical. So the oxidative stress is present inside the phagocytic cell. So by having this melanin productions, they can overcome the phagocytic effect of the human host. So the last one is the biofilm formation. So biofilm formation, this is just like our bacteria, it's a few layers of cell attaching to a surface and protecting fungal cell within the biofilm. So the structure here, we have a protective extracellular matrix. So biofilm formation is commonly found on mucosa surfaces and also endowing devices such as and prosthetic valves. So this is problematic for individuals who have all these devices in their body. The biofilm formation, not just bacterial biofilm, but also fungal infection can occur, but these mostly are candida species. And also biofilm can resist the antifungal treatments and immune clearance. For example, candida albicans can form dense biofilm with mixed yeast and hyphae cell, and these metric components reduce the drug penetration. So not just the immune to help the fungus being targeted by the immune cell, but also protect them from the antifungal drugs. So if you very quickly, these are the 6 virulence factors. I think the overview is here. The morphological transformation, switching from mold to yeast depending on the host niches. Environmental molds, when they're in the human host, they can switch to the yeast form. Yeast form can help them to be, one thing is they're less recognized by immune cell by changing because in mold structure, mold group, there are more beta glucans as compared to yeast cell. Beta glucan is one component inside the cell wall structure of fungi. So by switching to the yeast form, their last beta-glucans are being recognized. So in that way, the transformation from from mold to yeast protect the host. But if we talk about yeast that can switch to mold forms such as Candida albicans, the hyphae structures help the Candida albicans to invade and spread to other parts of the body. So it can go either way, mold to yeast, yeast to mold. That's what we call the dimorphism. So this is the first virulence factors. And metabolic switching, depending on where they are, they can acquire different nutrition. For example, if they cannot get sufficient glucose in Candida albicans, they can switch to the form of lactase, use lactase instead of glucose. And if they require ions, just like bacteria, they also require ions, they can produce and upregulate the siderophores, for example, aspergillosis, to help the fungus to acquire ions from the human's host cell. And adhesins help the fungus to attach to the host cell for colonization. Secretion of different type of enzyme to help them to invade or degrade, for example, immunoglobulin antibody, for example, catalase, superoxidase to help them to overcome the oxidative stress in the human host cell, for instance, the macrophages or phagocytic cell. They can also produce virulence factors such as alpha-1 triglucan that protect beta-glucans, which is being targeted by dectin, the immune receptors. And they can also produce melanin that will help them to resist oxidative damage within the phagocytic cell. Biofilm formation, just like bacterial cell, they form biofilm. They can resist immune response in host. They can also protect the fungal cell within the biofilm from being targeted by the drugs because there's less penetration of antifungals across the extracellular matrix of the biofilm. So these are the 6 virulence factors that we see in the fungal cell. So if we do a quick recap or a quiz questions, which of the following virulence factors enables the Candida albicans to invade host tissue and evade immune responses? Would it be A, B, C, or D? So this one I repeated a few times just now. A, B, C, or D? 2 answers. What about the rest? B or C? What about the rest of you? So Candida albicans is a yeast. So it is one of the unique Candida that can transform into mold structures. So beta-glucan synthesis is not part of the formation, the virulence factors of Candida albicans. So most of you pick B, one of you pick C. So the dimorphic switching from yeast to hyphae, the yeast to mold, applies to Candida albicans. So as I mentioned just now, the hyphae formation or the mold growth stage can help to invade the host tissue and help them to invade other tissues, as what we see in Candida albicans. Production and aflatoxin doesn't apply to Candida. So aflatoxin is a group of toxins that mostly produced by environmental fungus, which is not in Candida. So endospore formation is not correct. Endospore formation is present in bacteria. So the answer here would be B, switching yeast to hyphae structures. If we go to the next one, I think there's one more question. Melanin in fungal pathogens such as Aspergillus contributes to virulence by promoting adhesions, enhance nutrient uptake, scavenging free radicals and resisting oxidative damage, facilitating antifungal drugs uptake. So you see C, it cannot be D, okay? none of them, under normal circumstances, they will not want to take in more antifungal because antifungal are supposed to kill or inhibit the fungal growth. So the answer would be C, scavenging free radicals and resisting oxidative damage. So many of these environmental fungus species, they produces melanin, mainly for oxidative stress. So all good. I will then move on to antifungal chemotherapy. Any questions so far? All good. Who haven't checked in yet? All good. The code just now was, what's the code again? Maria. Okay, who else haven't checked in? Maria is here. Maria. Okay. Check in, Adeline. 84 check in already. It's a few students. Okay, let me come back here. Where are the rest? Working on assignment, is it? Okay, let me go to the next one. Antifungals. Yeah, let me just, I don't want my face to pop up later in the recording. I find it a bit distracting. Okay, so let's move on to antifungal. So antifungal chemotherapy, because just remember that fungal cells, they're also eukaryotic cells. So because of that, human cells are also eukaryotic cells. There are many similar molecular structures that's present in fungal species and also human. And for this reason, there are less drugs, antifungals, as compared to antibiotics. So these antifungals, because if they target a specific molecules or molecular structure that's present in eukaryotic cells such as fungi, most likely they will be present in human cells as well. So this antifungal, we must make sure that it's not toxic. So as put in here, because of the similar cellular and molecular processes, they make it difficult or more challenging to find new antifungal that are safe for human consumption as well. Because of the similar cellular and molecular processes and a lot of homology among genes and proteins, selection of antifungal remains far behind compared to antibiotics. The other reason that we have less antifungal is also because fungal cells, as compared to bacteria, they grow much slower. And they're often present in a multicellular form, such as mold, and that makes it more difficult to quantify. For example, how do we then determine what is a concentration that's effective against fungals to take effect? So they are not unicellular cell. Yes, yeast it is, but most of the mold they are not. So they grow slower. It is more difficult to come to a conclusive outcome to say that okay, agent A is effective, agent B is not. And because of the slow growing and often the multicellular form, it can then also complicate the experimental designs that are used to evaluate the in vitro or in vivo properties of potential antifungal agents. The reason being eukaryotic and slow growth in multicellular form makes it more difficult and less antifungal medication available today. So if you look at the antifungal chemotherapy, there are only 6 classes of the antifungal. So the orders of these antifungals differ slightly from the next few slides. I rearranged this to make it easier to see what are the commonality among the 6. For those of you who are in the morning tutorial, I'll mention very quickly. Paulines, that includes amphotericin B and nystatin, is one group. Terbinafine and also azoles. These 3 groups of antifungals, paulines, terbinafine, and azoles, they affect the ergosterol, whether if they bind to the ergosterol or they inhibit ergosterol synthesis. We also have flow cytosine, we have echinocandins, and grisosulfin. So only 6 classes of antifungal. So just a very quick overview of the fungal cell wall structures. So in the fungal cell wall, they have also the cell wall structures that's mainly beta-1,3 and beta-1,6 glucans. So remember just now when I talk about the virulence factors, we say the beta-glucans that's present in cell wall. Okay, so and also the other important one will be the ergosterol structures that's in the membrane, the orangey color structure. So this is also a lipid structures. So compared to human cell, we have cholesterol compared to ergosterol. So there's a slight difference for fungal and human cell. So ergosterol is the target of 3 different groups of antifungal. And if you look at the beta-1,3 glucan synthase, this is also another molecular structures that target by antifungals, and then the beta-glucan structures. So these are the 3 main ones that will then later on refer to this. So the first group of paulins, antifungals paulins, include amphotericin B and nystatin. Why are they separated into 2? So they are actually quite similar in terms of mechanism of action, but the application, the use of the antifungals are quite different. Amphotericin Bs are usually used for systemic mycosis, meaning that the fungus or the yeast are growing inside different part of the body. So we use amphotericin B. But if it is at fungal mycosis that's localized in certain parts of the body, nystatin can be used. So amphotericin B and nystatin, all the paulins, they have a cidal effect, meaning they can kill the fungus cell rather than inhibiting growth. So as shown here, they're most effective drugs for severe systemic mycosis, usually given intravenously. So they are generally broad spectrum. Amphotericin B can kill many types of fungal species. And also the resistance to amphotericin B is quite rare, which is a good news. But amphotericin B is quite toxic, and that's why they're only reserved for severe mycosis such as systemic mycosis. So in terms of the mechanism of actions, they form complexes with ergosterol, so bind to ergosterol and lead to the leakage of membrane and damage to the cell. So if you recall what we are seeing here, we have ergosterol. Okay, so the paulins such as amphotericin B and nystatin will bind to the ergosterol and form a complex. So when there's a complex formation, the structures of the membrane is no longer smooth structures. It affects the integrity of the cell membrane and causes leakage of the cytoplasm content. So as a result, the fungal cell will die. For nystatin, as I mentioned just now, is a topical used as cream or can be taken as an oral drug. So they're commonly used to treat local candida infections of the mouth and vagina, and they're used to suppress the subclinical esophageal candidiasis and the GI overgrowth of candida. So these are not the very severe mycosis. This is more mucosal and cutaneous mycosis. So similar mechanism of actions to our amphotericin B. And there's no systemic absorptions, so systemic mycosis should not be used. No side effect, but there's no effects against dermatophytes. Flow cytosins is another group of antifungal. So if you look at the structures here, the flow cytosine, they are an analog for the pyrimidine. So with the addition of the fluorine on the structures, it is an anti-metabolite. So it competes with cytosine that is used in the formation of RNA, DNA. And the flow cytosine penetrates well into tissue, including the cerebrospinal fluid. It is used as an oral antifungal and can be used in conjunction with amphotericin B to treat different mycosis such as cryptococcosis and also candidiasis. This is a combination drug. And they are also effective against the dermatiaceous fungal infections. So these are mainly the subcutaneous mycosis. So this is the second one. a pyrimidine analog, and it acts as an anti-metabolite and inhibits the DNA RNA synthesis. It competes with the cytosine. And we also have big groups of azoles. So the first group of azoles, such as the triazoles, there's no need to memorize all these names, but I group them separately to show you the differences in terms of the application. So the first group of azoles, which are the triazoles, they are commonly used as an oral or IV drugs, used for systemic and localized fungal infection. So the application is quite broad. The mechanism of actions for azole is that interfere with the synthesis of ergosterol. So if you recall what we talked about paulines, right? Pauline binds to ergosterol. Pauline binds the structure ergosterol directly, but our azoles doesn't bind to ergosterol. It inhibits the fungal cytochrome P45 enzyme. So when the enzyme activity is inhibited, it interferes with the ergosterol synthesis. It interferes with the conversion of elanosterol to ergosterol. Let me just skip to slide number 24 to show you what this is. So if you look at the formation of ergosterol, that forms part of the cell membrane structures, paulines such as amphotericin B and nystatin binds ergosterol, leading to the leakage of membrane. But our azoles, they bind to the P450 enzyme that interfere the conversion of elanosterol, which is what we see in the pathway of the formation of ergosterol. This is where it is targeting this enzyme. Okay, we also have another group of antifungal called terbinafine. So these, as I mentioned earlier, there are three different groups of antifungals targeting ergosterol, whether it's a synthesis or it's a binding. Terbinafine, let me find terbinafine. Okay, so terbinafine targets the earlier step of the ergosterol synthesis. It binds and inhibits the squalene epoxidase enzyme. So by inhibiting this squalene epoxidase enzyme, it leads to the squalene accumulation. So squalene is actually required to form elanosterol and later on transform into ergosterol. So the mechanism of actions of the terbinafines then affect different steps of the ergosterol synthesis. So here, terbinafine, azoles, and paulines. Okay, so we have already talked about before cytosine. This is anti-metabolite, very similar to the pyrimidine structures. We talk about azoles, echinocandins. So examples of echinocandins include caspofungin. So what it does is they will affect the synthesis of the cell wall polysaccharide beta-glucan synthase and disrupt the cell wall integrity. So this is what I mentioned just now, the beta-glucan synthase. So this is targeted by echinocandins. Okay, and the last group of antifungals is our graciofulvin. So graciofulvin is commonly used for dermatophytes. And what happens is once the graciofulvin is absorbed, it becomes keratinophilic, meaning that they will accumulate on the keratin. Keratin that form part of the human skin structures. They will accumulate in the keratin. That's what we call the keratinophilic. Concentrate in the stratum corneum, which is the outermost layer of skin structure, and inhibit the hyphae form growth structures. Remember last week when we talked about dermatophytes, they're actually caused by fungal species. So this graciofulvin accumulates on the outermost layer, which is the target structures of our dermatophytes, and inhibit the hyphae growth. So the mechanism of the graciofulvin is actually interact with the microtubules and affects the mitotic spindle formation. So this is required for the cell division. So by inhibiting and disrupting the mitotic spindle function, it inhibits the growth of the hyphae and the fungus. So if you look at the 6 different groups of antifungals, here, the first 3 targets the ergosterol synthesis, which is part of the cell membrane structures. Flow cytosine is a pyrimidine analog, so it interferes with RNA DNA synthesis. Echinocandin interfere with the beta-glucan structure formation, which is part of the cell wall. Raciovalvin then affects the mitotic spindle function, disrupt the mitotic spindle function, inhibiting the growth of the hyphae and the fungus. 6 different groups, classes of antifungal. So what I want you to do is, if you have the habit of making your own notes, just quickly write down the different mechanism of action that will help you in understanding the different classes of antifungal. So if you look at the question, which of the following antifungals acts by binding to ergosterol and forming pores in the fungal cell membrane? So we have caspofungin, this is echinocandin. We have amphotericin B, part of the polys. We have fluconazole, which is azole, and we also have flowcytosine. Amphotericin B. Amphotericin B, huh? Okay. What about the, can you recall, if I ask you to just quickly type the mechanism of action of your azoles, so-called azoles, how does this antifungal work? Anyone? So bind to the P450 enzyme. Then what happens? What is the outcome? Inhibit ergosterol conversion. Is it the inhibit the ergosterol conversion? Inhibits the ergosterol synthesis, correct? By let me see. By preventing the conversion of lanesterol to ergosterol, that's correct. So how about terbinafine? What about terbinafine? What is the mechanism of action of terbinafine? Terbinafine is also targeting the ergosterol, right? Is it the binding to ergosterol or inhibiting ergosterol synthesis? Inhibits conversion to lanesterol, which is then converted into your ergosterol. Okay, that's correct. I think you all remember already. Very good. Very good. Next one. Hey, what about the rest of you? Why so quiet? Only Wizen and Jessica. And Chicken. No, Chicken didn't answer. Chicken was saying. Okay. What about the rest of you? Which antifungal drugs inhibit fungal DNA and RNA synthesis as a pyrimidine analog? Not terbinafine, right? We just talked about terbinafine. Not azoles. So C or D? C. What about the mechanism of action of grisovalvin? Giselle okay? Yutong okay? Wizen okay? What about the rest of you? Are you all following? Grisovalvin. What does it do? What is the mechanism of action? Hello, anyone there? A different individual who answers this, please. And let me see who are here. But how? The one wasn't ready. Another person. Disrupt mitotic spindle function, okay? What does it mean? What does it mean as it disrupts the mitotic spindle function? Inhibit growth, okay? That's the outcome. Inhibit growth from growth. Do you think that this is a sidal or static effect? Is it inhibiting growth or is it killing fungus? Okay, answer is C. What about glycosylphosphate? Static, right? So this is inhibiting growth. This is not killing effect. So for glycosylphosphate, the effect is actually fungal static, not sidal, unlike our pollens that can kill the fungal cell. The rest of you all following? Yes? Can we take a 5 minutes break and then we'll start again? Okay? Rest for a little while. 5 minutes. So we'll start at 12:55 again. I will pause the recording.